Hey everybody, it's 8 o'clock, so we have a new live stream coming at you for AP Biology. So I see a couple of you are already here. Welcome. Oh, we've got 5 people. Great. So I'll kind of start things off slow to let everybody get here and get logged in. So go ahead and write in the chat box where you are streaming in from tonight, just so we can kind of see across the country where everyone's coming in from. I'll type in here as well. Coming at you from Michigan. Right now it is raining like crazy, so you could probably maybe even hear it. Hopefully my sound is okay tonight. Got Massachusetts. Hi, Elena. Or is it Elena? Sorry. California, great. So everyone's just joining. We'll give everyone a couple minutes before we really get started. I like to see where everyone's coming from. It also gives me an idea. Virginia, hi Sandra. Gives me an idea if this is a good time to be streaming, if 8 o'clock is too late or too early. California, does that work for you okay, Javier? I know it's 5 o'clock your time. It's probably just before dinner, which is kind of crazy because in Michigan it's 8 o'clock right now. You like it to be later at night? Okay, that's good to know. Yeah, 5 o'clock's really early. I have a really good friend who lives in Los Angeles, and it's always amazing what that 3-hour time difference can do. So yeah, maybe we'll do some AP Biology streams later in the evening. Okay, usually catch the replay. Oh, okay, nice. Yeah, replays are great too. So if you're on the Fiveable Plus account, catching the replays is amazing. So great. Well, anyway, I'm Kari Parnan. I'm just going to be running a Unit 1 review for you. Your teacher might not call what you've done Unit 1, Unit 2, kind of how we're running it with Fiveable. But basically, our Unit 1 is modeled after the College Board. So we're going to just be going over biochemistry. So if you've seen any of our live streams over the last couple of weeks, it's just the culmination of all of that. Can I have everyone just write in here what unit you're currently on in AP Biology? So me and my team can get an idea of where you're at, because maybe you're really far ahead of us and we need to pick up the pace. Unit 2. Okay, some people are in evolution. You're still on Unit 1. Jesse, what is your Unit 3? What do you guys call Unit 3? What are some topics you're doing? Okay. Cellular respiration. Okay, so like cell energetics. That's good. So we will definitely be getting to cell respiration soon, so sit tight. I know that's a tough subject, so we'll definitely be coming at you with some good streams. Evolution, okay. Some teachers like to start the year with evolution. I think that's really cool. We're just modeling it from the AP College Board, but whatever we hit, you'll hit as well. All right, Tucker says he's the same as Peyton. Peyton, you're self-studying. Okay, cool. Awesome. So this will be a really good resource to be logging in and checking out some things. Excellent. Okay, good to know. So some of you, at least for biochemistry, if you're not in this unit, it's so good to go over this stuff, especially at the beginning of the year. So much of it seems familiar as far as you all took chemistry before. But sometimes you almost get overconfident. Like, I get the chemistry stuff, no problem. And then come May, it's been almost 2 years since you took a real chemistry class. And then you're sitting there trying to explain polar covalent bonds or, the polarity of water, and you kind of get lost. And you know the word polar, and you should be able to explain it, but maybe you're not quite there. So anyway, thanks for joining, everyone. It's really good to have you here. Before I officially get into the content, if you haven't already, follow Think Fiveable on social media, and you can also follow me. I'll show you that at the very end, what my Twitter account is, because then you can find out exactly what's going on. I'm sure a lot of you are in other AP classes too. So if you follow Think Fiveable, you'll know exactly when things are going to be streaming and get a schedule, and then you can kind of make sure you log in on stuff that you need. This week, I'll be doing 2 live streams, Tuesday and Thursday night. all about cells. So we'll look at the subcellular components, cell structure and function, and cell size. So for those of you who are in Unit 2, I'm assuming, like Teresa, you said you were in Unit 2, so I'm assuming that's all like cell basics, hopefully. So we're going to get into that this week and then kind of gear up, and it's just going to get faster and faster. It'll be really fun. Tonight, though, is all review on biochemistry. So I kind of made a review guide for you that if you are the 5able Plus, if you're on the 5able Plus plan where you can look back at replays, I'll put the actual PDF document of the review up so you can print it at home. But for now, I just put it into my slides and we can kind of just go question by question. So that's what we'll be doing. All right. So first, let's just look at this. This is a list straight from the College Board of key terms from Unit 1. So I'm going to give you about 30 seconds to look at that list. And then type in the chat box 3 words that are least familiar to you. Or 3 words from this list that you're like, I could use a little bit of extra help on that. So I'll give you about 30 seconds. I'll set my timer and then come up with 3 of those words that you feel like you could use some extra help on. All right, and then I'll just get like a little list going here. Everything that isn't related to chemistry. Okay, cool. Okay, got some cohesion and adhesion people. Yeah. Okay, cohesion, adhesion. Phospholipid, very good. Oh, good, the type of bonds. Thank you, Jesse. Peptide bond and sugar phosphate, love it. Those are good things to go over. Okay, semi-conservative. Polysaccharide, phospholipid, okay, good. Bonds, okay, cool, Jesse, we'll do some work with bonds. Polysaccharide, monosaccharide. All right, cool. Polysaccharide, monosaccharide. TYSM. Does that mean thank you so much? Alpha and beta. Okay, good. We will hit that, Javier. We'll do alpha and beta. I've got a practice question, actually, that hits on that really well, so that should help. Okay. Bonds. Okay, got it, Jed. So there's a lot of bonds. You don't remember which one's which. Okay. All right, cool. I think I heard from everyone. Okay, so what I wrote down, I kind of just made a checklist as you were all typing. We've got a lot of people wanting to look at semi-conservative, replication, water stuff like cohesion and adhesion. They're super similar words, so definitely that could be something that's confusing. Peptide bonds, sugar phosphate bonds, bonds in general. So ideally, I think what we can do is look at where do we see the type of bonds, because then that'll help concrete it. Like if I'm talking about a sugar phosphate bond, where is this even happening in biology and why is it important? So we'll get to each of those. And then polysaccharide, monosaccharide. So we'll get to those. That sounds really good. Thanks, everybody. Okay, so actually, I'm just going to see if I can make a brand new slide based on what you guys all told me. All right, let's see here. Whoops. Give me 1 second. All right, let's just start with semi-conservative replication because so many of you had that as your number 1. So first of all, that has to do with the replication of DNA. Conservative is a word in biology. Functional groups, sure. Let me write that down so I don't forget functional groups. So conservative means how consistent something is. So if we were to say its replication is fully conservative, that would mean your DNA molecule basically just replicates and your new molecule is 2 new strands and your old molecule is the 2 old strands. So you might have seen a teacher, and I'm going to do kind of try my best on a drawing here. Okay, so hold on, I'm gonna try this. This is gonna be really not tech savvy, but okay. I've got a red double-stranded thing of DNA. Okay, so when we say semi-conservative replication, what we mean is when this replicates and I get a brand new strand, it's gonna be half old and half new. And I'll explain what I mean by that. So this red strand is gonna split into two. It's a little more complicated than that, but when we get into DNA a little bit more, we'll go really in depth. But the idea is your original parent strand, okay, where I showed it in red right here, okay, that starts to split down the middle. We get a lot of enzymes that help us do that, but then new nucleotides are added to match complementary with that. So like if this one is a G, we're going to get a new C added in. If this one's an A, yep, good, Javier, hydrogen bonds break, right? Because the middle of your DNA molecule, like the rungs of your ladder, the horizontal aspect, those are your bases held together by hydrogen bonds. So this starts to split, the hydrogen bonds break, and so those individual bases, let's say it was a T and an A, those are now exposed, okay, we've broken the hydrogen bond. So we have polymerases, which we'll get way more into depth later, but the basic idea is you get new nucleotides added, right? Remember, a nucleotide is a phosphate sugar base, okay? So we've got a phosphate and a sugar, and then that base is going to be complementary. So these two strands, these are going to be my new strands now. So it's 50% red and 50% green. This is just the more specific term for DNA replication, yes. So semi-conservative means it's not going to be like two parents, a full parent strand and a full new strand. It's half and half, right? So this original strand was from the parent DNA, and this one was also, the red was from parent DNA. They did a bunch of complicated experiments to figure that out, where they basically tagged the phosphate sugar backbone with something, and so then they were able to see with the new two strands that, hey, both were glowing, and they tagged them with something that was that could glow. But that's what semi-conservative replication means. It's an explanation for DNA replication where it's not just one parent becomes, it splits, makes new strands, and then the parent finds each other, and the new two strands find each other. It's half and half, but it's still two identical molecules. Okay, so that's semi-conservative. And the reason why that kind of matters and ties into this unit, because this unit isn't all about DNA, the reason it ties in, one, to think about hydrogen bonds breaking. So thank you, Javier, for bringing that up. Your hydrogen bonds are the ones that have to open up, but also the idea of knowing the structure of DNA as an anti-parallel structure. So where you have 5 to 3' on this end, and it runs 3 to 5'. It's basically upside down on the other end. And the reason it runs that way is that so when we do start to split it and add nucleotides, then we just have two brand new DNA molecules where you don't have to go back to the parent and rejoin the original. So without that 5 to 3' anti-parallelness, DNA replication would be a huge, huge mess. Okay, I'm going to try to skip ahead. Let's see here. If I go to properties of water, I'm going to skip even more. Sorry about this. This is kind of from an old PowerPoint, but this is another one that someone brought up, cohesion and adhesion. So if you think about what the word co means, like when your teacher tells you to collaborate or cooperate, right? It's talking about like togetherness in a way. And so for co, we're talking about water binding with water. It's togetherness between water molecules. So if you did the penny dropper activity at school, possibly, where you put water droplets onto a penny and you're amazed by how much actually fits in there, a lot of that is attributing to water molecules sticking together. They stick to each other because of hydrogen bonds. Yes, Jesse. Yeah, Jesse just said that learning prefixes and suffixes helps in AP biology. Absolutely. I completely agree. And if you're going to eventually take anatomy, prefixes and suffixes are great. It helps you dissect certain words, right? Especially we talk about like hydrolysis, dehydration, intermolecular forces. So inter versus intra, is that something you have a question about, Tucker? The difference between inter and intra forces? Oh, you had a flashback from chem. Nice. Yeah, but I mean, it's perfect for water, right? Because if we're talking about water, the bonds that are holding together the actual water molecule is an intermolecular force, but intra would be the water molecules together. So cohesion is actually an example of that intramolecular force. It's going to be between other molecules. Adhesion has to do with water bonding to another material, right? So like water sticking to some other substance. So in the picture here, I have water droplets like sticking to the side of this plant. In reality, that doesn't serve a huge function necessarily, but within the plant, when we have vascular plants that have xylem and phloem, and material needs to go from root to shoot, so from like the roots of a tree, and let's say you've got these huge redwoods in California, right? My California person, water has to get from the ground all the way up to the highest leaf. And the reason it does that is water sticks together through cohesion, but then it's also being pulled up and it's sticking to the sides of these vessels that help it creep up to the top of that plant. So cohesion and adhesion there. What was another one people had a question about? Certain bond types. So peptide bonds. Well, let's go to a quick picture of a water molecule. Hold on. Sorry, I'm kind of popping around everywhere. Okay, so here's a good example where we have two types of bonds happening in just this picture. Okay, so I've got two water molecules pictured here. Okay, the type of bond between this water molecule, so I have one oxygen and two hydrogens, that's going to be a polar covalent bond. So just between that one water molecule, it's oxygen to hydrogen, that's a covalent bond. Covalent meaning they're sharing it. Okay, they're sharing the electrons. Water's unique and polar because it's unevenly sharing it, right? And so that's why we have that delta negative. So it's a partial negative charge. Hydrogens are partial positive, right? And because of those partial charges, that's where an example of hydrogen bonding happens. Okay, so covalent bonding, we usually show it with like a nice straight line to show it's a true bond. It's true sharing of electrons. But then the hydrogen bonds I like to draw them like this too, where they're kind of like a dashed line. It's more of like an electrical attraction. That's why I showed electrical or magnetic. That's why I showed the pictures of the magnets there. So oxygen, partially negative because it's so electronegative. It likes to hog the electrons that it's sharing. It's going to be attracted to, opposites attract, remember that. It's going to be attracted to the partial positive of this other water molecule, the hydrogen on that water molecule. So that's a hydrogen bond. It's not always between oxygen and hydrogen though, okay? It can be between nitrogen and hydrogen. It can be between carbons and hydrogens. Anything that's electronegative, that's going to be unequally sharing electrons or showing a partial negative charge, it can have a hydrogen bond with water, okay? It's weaker than a covalent bond. Good, fluorine, yeah, good. Good, yeah, so that's an example of our hydrogen bond, okay? And then if I were to skip ahead to, let's see here, I'm gonna try to find, so I should have all my things titled. Here we go, a protein, okay? Here's where we see a peptide bond. Okay, think P for protein, peptide bond. It's the bond between your individual amino acids. So when we go through that process of amino acid 1, the monomer, joining with amino acid 2, and we get a whole string of them, the bond between each individual amino acid is your peptide bond. Okay, it is a covalent bond, so technically we could call it a covalent peptide bond. So it's still sharing of electrons, but we call it a peptide bond specifically. Okay, so that is the original bond between that primary structure of amino acids. Your teacher might have talked about it like beads on a string. Like you can imagine each amino acid is like a circle, right? And they all just kind of join together, and the thing that's holding it together is peptide bonds. Then we start to get the other kind of intermolecular forces between all the different other amino acids. So I can find a picture of that. Where did I put that one? I'm taking a guess here. Hold on, sorry. My slides aren't all numbered, so I'm jumping ahead. And sorry. I keep going until I find my slide. Here we go. This will be our next slide. That'll help for those of you who were wondering about alpha and beta sheets. So here it's showing like beads on a string, okay? So I've got primary structure. I know the picture is kind of small, but I didn't want you to necessarily need to read these words, but each of these circles is an amino acid, okay? So it's numbered A1 through A11, I think. Haha. Yeah, Jed, I'll talk to you about that in a second. That's very true. But yeah, we've got these beads on a string held together by peptide bonds. Remember, think peptide, primary structure, protein. Okay, so those are all P things, right? Protein, primary structure, peptide bonds. Okay, the secondary structure, when our protein starts to fold, you can get alpha and beta helix. And so that's actually just hydrogen bonding between the amino acids themselves. So I know you think back to maybe the amino acid R group and how unique that is and how that'll contribute to bonding. And it definitely does, but the just basic hydrogen bonding can happen just straight between these amino acid side groups. If we were to go back to looking at the basics of an amino acid, right? Each one has like a carboxylic acid side, and the other one has an amine side, NH3, right? So that's why it's called an amino acid. Amine, the N group, acid from carboxylic acid. Each amino acid has both ends, right? It has an acidic side and an amine side. And so those chains can form alpha helix and beta sheets. Okay, hopefully, does that answer your question about alpha helix and beta sheets? Who asked that, Javier? Yeah. So that's the secondary level, Javier, of folding for proteins. And then the tertiary structures tend to get more strong, where we get like ionic bonds between your positive and negative amino acid R groups. You get a lot more hydrophobic and hydrophilic folding. The phobic ones go in the middle. The philic ones, they love water, so they're on the outside. And then that fourth layer for your protein is going to be when you have multiple units coming together. Like, so for hemoglobin, for example, it's actually four sequences finally bound together. And so it's a quaternary structure. There's four. And so you get 4 subunits. It's this big globular protein. Not all proteins are like that, but that's the fourth level that you could get to. Yeah, Jed made a good comment about how a lot of the macromolecules there's definitely a covalent bond, right? Because remember, they're all linked by dehydration synthesis. So water's been taken out. And so those other things are going to bond together. They want to share electrons. So yes, absolutely. They're covalent bonds, but there's something different, right? So with carbohydrates, we call it a glycosidic linkage, proteins, peptide bonds, all that stuff. But try not to worry about the terminology too much. As long as you can kind of structurally and conceptually explain, is usually helpful. Okay, someone just asked, what is folding the amino acids? The amino acids are folding. So if you think of like this beads on a string, the actual circles that they're modeling, they're not actually circles, they're an actual amino acid. But each of those amino acids, it's not just going to stay a bead on a string, because what we're building is a protein. And a protein has a job, right? And a protein, to do its job, has to be a certain shape. I think of it like how, you know, the key to get into my house is different from the key to get into my car and to get into school, right? Like where I teach, I have to use like a little card and that scans me in. It's still a key, it's still serving a function, but its shape is very different. And if I tried to use my car key to get into my house, it wouldn't work. And so with proteins, it's the same thing. It's that whole lock and key method where every protein has a shape. And if its shape is different than it should be, it is not going to be able to do its job. It's not going to be recognized by other molecules. It's not going to be able to function like it needs to. So to answer your question, the amino acids start to fold in on one another, and then their final shape makes it the fully functional protein. Okay, it's not going to be functional if it's just in that primary structure forever. It has to start folding immediately, and that's why if there is a mistake, you know, if one hydrophobic amino acid becomes hydrophilic, or one acidic becomes basic, the whole thing could potentially not work anymore. And that's where a lot of our diseases come from, right? Like the sickle cell anemia comes from one incorrect protein, or one incorrect amino acid. Okay. And that's it. Just one little amino acid is wrong, and it made the whole protein behave differently, causing a disease. Okay. All right. So we've gone over bonds. We've gone over semi-conservative replication, a little bit about water, phospholipids, all that stuff. For the person who asks about functional groups, we're going to kind of tie those in as we go once I start looking at some practice problems with everyone. And so within each practice problem, we'll say, okay, well, where's this carboxylic acid or carbonyl group? Where's our hydroxyl group? And we'll kind of look at those individually. All right, so if we go back to our, here we go. Okay, so this is another one I wanted to show you. Again, this will be on the PDF. So you can draw it for yourself real quick or just kind of take a look at it and think about what you'd be able to fill in on your own. This is just a concept map from many years ago that I made. And again, it'll be on the PDF. You can download it later if you're a 500 plus person. Otherwise, you have access to it right now, and you could take a picture or whatever. But anyway, this is just macromolecules. If you're able to fill this in, it'd be really helpful. So if we look at just the top, it's breaking it down off to the right with 4 major types. So Peyton, there's going to be a link to the PowerPoint once it's all uploaded to 500. So if you're a 500 plus, you'll be able to grab access to the PowerPoint. Good question. But I mean, if you're not a 500 plus person, you're more than welcome to to snap a picture of certain screens if there's something you want. So I'll stay on this one for a little while. But anyway, I think concept maps are great. You can even make your own, and ideally, when you make your own concept maps, your brain works differently than mine. So I'm very linear with things. I like things in lists. You might be someone who's more artistic and expansive. And so you might like to have something in the middle and then branching off from there. But it's a really nice story to be able to walk yourself through and say, okay, do I know the function? Do I know the structure? Do I know how they're made? Do I know how they're broken down? And if you're able to fill something in that was blank originally, that tells you a whole lot more than just flipping through your notes. And I think with working with students over the last 8 years, I've noticed a lot of students just reread their notes or they rewrite their notes. And they spend hours rewriting and making everything look really pretty. And they think they know it because they've written it down so many times, but then they get to a test and there's a free response question and it's asking them to link structure and function of such and such molecule. And they're like, oh, I have no idea. So just keep in mind that look at a blank canvas, and if you can fill in a blank canvas without looking at your notes, you know the material really, really well. Okay, so if we were to fill this in, you might wonder, okay, well, how do I know which macromolecule goes where? You can kind of look down and start to fill it in. So for example, the only one that gives us a clear idea is the second one that has fats and phospholipids and steroids as the example. The other ones, we're not really sure so far. So I'm going to click through and at least help you fill out this first part. So what is it formed by? This is something we should all know by now. Macromolecules are formed by dehydration, or you can call it condensation, same idea. Dehydration synthesis, which releases water, right? Dehydrate, water is taken out, right? So an OH group, a hydroxyl group from one monomer and a hydrogen from another come together. So Javier said, could monomers work? It could, but I have the letters RX there. That means reaction. So they are formed by monomers, absolutely. Yeah, absolutely. But that RX means reaction. But you're not wrong. They're absolutely formed by monomers, which releases water, and then they're broken down by hydrolysis reactions, which require water, right? Hydrolysis, lyse or cut with water. Okay, so water comes in, breaks those bonds. It gives one group a hydroxyl back, and it gives the other group a hydrogen back, and then everything's nice and balanced. Okay, and then I'll fill in the top 4 for you as far as our macromolecules. So we're going to start with carbs, lipids, proteins, and nucleic acids. So ideally, as far as structure goes, on each of these, I wouldn't expect you to be able to draw a perfect, glucose or fructose monomer, right? Like you're not going to be asked to draw that, but have a general idea of what elements are present in each of these, right? So, and also, have an idea of, okay, well, if I'm talking about a lipid, I know there's fatty acids in there, which is a really long hydrocarbon chain. So there's lots of carbons, lots of hydrogens. Proteins, And nucleic acids are going to be our only ones with nitrogen, right? So maybe you're not going to have to memorize these structures in terms of drawing them yourself. You know, the AP College Board is not going to say, you know, draw me a perfect nucleotide, but it might ask you to compare the elements found in a nucleotide compared to the elements found in a carbohydrate. And you'd have to talk yourself through, okay, well, carbohydrates are mostly carbon, hydrogen, oxygen. Each carbon likes to have 4 bonds. So if I'm bond to 2 carbons, then what else is coming here? It's probably 2 hydrogens. So there's lots of hydrogen. And so just kind of being able to talk yourself through that and walk yourself through it is really helpful. Okay. So I apologize for the bad handwriting here. But structure, you would want to know the monomer is a monosaccharide. Mono meaning one, saccharide meaning sugar. So your teacher might have even just said the monomer of carbs is sugar, and that's okay too. Elements present, CHO. It can either be in rings or a long chain. Not every monosaccharide is going to be in a ring. And then function is energy storage. If you want, you can go back to my other live stream where I really went into each one and how structure fits function, and how even within the carbohydrates, the idea of like cellulose versus starch and glycogen, those are concrete examples of, well, look at this perfect structure. It's very branched compared to something that's like highly stacked like cellulose. Why do those structures let them do each of their jobs? Is the monomer the empirical formula? The empirical formula of like glucose, for example, where it's like C6H12O6. You could say that. You could just say the monomer is a form of C6H12O6. But the idea is, yeah, your carbons and oxygens are going to be a similar subscript, and then your hydrogens will be double that. So you could, think of it in terms of just numbers. Okay, moving on to lipids. We've got some examples here, or at least I only did 2 of them. So our first just normal lipid is going to be glycerol and fatty acids. We've got this hydrophilic head of glycerol and fatty acids. Sorry, that's a phospholipid. Long-term energy storage and insulation. A phospholipid, the second one, is going to be that hydrophilic, hydrophobic, and it's forming membranes. That'd be the most, I would say, if you're able to explain why a phospholipid can form membranes, you're going to be in a really good spot. Okay, so talk about how it's a double layer membrane. We'll get more into that with cells. I think there's like an entire day of cell membranes. Are lipids always linear? Well, the fatty acids are going to be pretty linear unless they're saturated or unsaturated. Okay, if they're unsaturated versus saturated, then you get those bends and kinks. But remember, another example of lipids, if I go to my next slide, are steroids. And so steroids can be in rings. So lipids are kind of a weird macromolecule in that, one, they actually don't form polymers. So their monomers are going to be glycerols and fatty acids, but they don't actually form polymers. You can't just do strings and strings of glycerol and fatty acids. So a glycerol is going to have 3 carbons, and so there's room for 3 fatty acids, or there's going to be room for 2 fatty acids and a phosphate, which is your phospholipid. So lipids are kind of weird, where they're not going to be these like really pretty structures. And the fact that steroids are linked into lipids as well. I wouldn't worry about trans fats. I would think you should be able to identify the difference between a saturated and an unsaturated fat. And so saturated means like fully covered in hydrogen. So that's where you have your fatty acids that are going to be straight because there's no double bonds in them. So it means all the carbons of that hydrocarbon chain, they're saturated with hydrogens. Okay. Unsaturated would mean that some of the carbons aren't fully saturated in hydrogens. And the way they get around that is that you've got carbons with double bonds, right? So a C next to a C with a double bond. And so if you're double bonded, you don't need those extra hydrogens. And so that double bond actually starts to bend. And when you get into organic chemistry in college, you can learn all about like cis and trans. in terms of bending, if it's going to be bending up or down or what kind of angle you're working with. But the idea is with an unsaturated fat, you're going to have a little bit more movement, which is really good thing, right? Depending on what kind of fat we're working with. We don't want to have a super solid fat, you know, in terms of like rock hard fat, if we're needing some sort of movement in an organism. And so, yeah, I would know saturated versus unsaturated. And remember that saturated just means fully covered or fully bonded to hydrogens. Unsaturated means double bonds, and that would give you your bend in your lipid. Okay, the other one I wrote in here was nucleic acids, of course. Monomer is a nucleotide. Function is coding for protein, or you could say it's the, you know, nucleic acids are the genetic code of life or something like that. It has something to do with heredity. All right. Okay. So again, that's just my example of a concept map you can make for yourself. And it's just about macromolecules. You could take any topic and just say like nucleic acids, go. Write as much as you can about it. Right? We have about 15 minutes left. And so I do have some practice problems. Do you guys want to get to those or do you have more questions? ATP is not part of the nucleic acid. So adenosine triphosphate is a different molecule. It does have phosphate in it, which is also present in nucleic acids, but it's a different molecule. But it's used all the time and it's super important. We'll talk about ATP a lot in the next unit. And then for those of you who are in cell respiration, we will definitely hit on ATP and the importance and structure of it. Okay. I really like all the questions, everybody. It's good. This is like college level stuff of coming to a review session with questions. My professors always offered in-person review sessions before a big test because, you know, a lot of college science classes, they have about 2 tests and then a final exam. And that was pretty much your whole grade. And you could go, and if you didn't have questions, if people just showed up expecting the professor to go over something, kind of like how I'm doing with materials, a college professor won't do that for the most part. At least at my university, they did not. They would stand up on the podium, and if no one had a question, they would leave. So yeah, I'm glad you guys came with questions. Do non-plus members get the PDF? No, I don't have a way to link that to you. But like I said, we're gonna go, I have snapshots of each question. So at the start of every question, if you really want to grab a picture. But I think to be a plus member, it's only $5 a month maybe, and you get access to like all the AP classes. So I would consider it. You know, it's cheaper than Netflix at least. And then you'd get all the resources for sure. And the replays, which will be really nice during AP review season. You'll be able to go back and go like, I really need help with photosynthesis, and then find everything. Oh, there's a deal going on right now. Okay. See, these are things I should know. I'm so bad at Twitter to know. Okay, Javier said there's a deal going on. I thought it was $5 a month, which is not bad at all to get all this free stuff that we're making. And at least everything I make is, I've authored it. Oh, 40 for the whole year. Okay, cool. Yeah, so yeah, go on the Fiveable website and look at it. Because, you know, we do, the people who work for Fiveable, you know, I spend hours and hours putting these worksheets together and doing the drawings myself. And so if I just gave all that away for free, it would be pretty tough, and I probably wouldn't come up with as good stuff. So, all right, let's go look at number 1. So if you were to be asked to construct a bar graph that displays the relative amounts of hydrogen, carbon, oxygen, and nitrogen in each of the 4 types of macromolecules, think about first how you would do that. Your graph might look different than someone else's, but that's why the word relative amounts is important. Okay, so I'm not asking you to necessarily give me a number. You don't have to tell me that some glucose molecules have, you know, 6 carbons, 12 hydrogens, and 6 oxygens. It's relative amounts, and then you're doing a full comparison across macromolecules. Sebastian, I can go back to any particular slide you'd want if you wanted to grab a picture of it, but I cannot post the like the full PowerPoint, but it will be posted to the linked video once you replay it, and then you'd be able to have that, no problem. Okay, so a question like this, just kind of take it in and start to just let your brain think about how you would draw this. First, it does tell you to do a bar graph. The idea is you're trying to convey, I do know the different things about carbs, lipids, proteins, nucleic acids, and the relative amounts of those three elements. All right, and so I'm going to show you my very, very rough version of this. Oh, good. Good. So Peyton made a good point. She said, well, carbs only have carbon, hydrogen, oxygen, right? So definitely no nitrogen. Don't put that as a bar for carbohydrates. Same thing with lipids, right? So lipids should not have a nitrogen bar at all. So you could show a lot. Another thing to consider is that compared to carbohydrates, lipids can have extremely long fatty acid chains. And remember, fatty acids are just carbon and hydrogen, right? So carbon, 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 carbon. Each carbon has two hydrogens, right? So there should be about double the amount of hydrogens than carbons, but compared to our carbohydrates, there might be more of each of those things. Okay, and I wish I would have been able to spend more time on this, but you guys came with such good questions. We only got like 10 more minutes. Again, this is something I sketched super fast, bad handwriting, all of the above. But relatively, right, you're in a carb, the amount of carbons and oxygens you have are the same, about the same. Where with lipids, there's not a ton of oxygen in there, right? Because lipids are going to be your again, mostly fatty acids. Your glycerols each have an oxygen. But other than that, it's not going to be a really oxygen-heavy molecule, which is also what makes it so water insoluble. Remember, think like if you have a salad dressing and you've got oil and water, those are going to be separate because they're made of fats and lipids separate themselves from water. They're not going to fully be soluble. And so that oxygen, which normally contributes to polarity, right? Your oxygen should be pretty low in a lipid. Okay, so that would be something that sets it apart. Protein, it's going to have a decent amount of nitrogen, right? Because every amino acid has a nitrogen group. And some R groups also have nitrogen. Nucleic acids, I added the P on for phosphate, just even though it didn't ask for it. I just thought that'd be a nice touch to also mention that would be the only one that has phosphate as well. Okay, so again, these are relative numbers. If someone were to go through and actually analyze every carbon, hydrogen, and oxygen, I might be off by a little bit, but again, you're trying to convey general knowledge. Okay. Okay, next question. This is something AP College Board would write. They're into, this year especially, getting everybody to think about how a model can represent a biological principle. And how there's a lot of different models that can convey one thing. Like we can draw DNA in 10 different ways, and each way is maybe trying to show us something different, okay? So pay attention to models and pay attention to how things are shown to you. So here it says there's 3 models of water. For each model, explain why which property or characteristic of water it best illustrates, and then for each property, describe the biological significance. So this is also something the AP College Board loves to do for AP Biology, is that it gives you like 3-step problems almost. So don't rush. It's not going to be one of those things from your honors biology class where you were like, oh, the answer is polarity. Boom, I'm done. And then the answer is cohesion and moving on, right? Like they want you to go really deep with your questions. Look at those bolded words because every bolded word is something new they're asking you to do. So you need to explain what property of water it's illustrating and then describe that property. Why do we care about that property, right? So those are like 2 key things here. So if we were looking at just model 1, okay, it's showing 5 molecules of water and then it's got arrows pointing. So take a second and think about what property of water is jumping out to you that this model is showing. And then type it in there. Okay, could be cohesion. Brady said he thinks it's hydrogen bonding. Yeah. Hydrogen bonding. Oh, polarity too, yeah. Okay. So I love the participation. It's great. So some of you are saying several things. This is also what I love about the AP Bio test, is that for most questions, there are 3 or 4 options for what you can say right. And sometimes they're only asking for 1. And so if you hit 1 of the 4, you get those points. It's amazing. It's really, really good. So yeah, I would say at first glance, my mind is looking at those partial charges and it's pointing to the dotted line. So my mind right away goes to hydrogen bonding. So I would personally choose hydrogen bonding and say, oh, due to the polarity of these molecules, maybe define what polarity is and point to those partial charges and say, okay, because of the polarity, there's attraction between opposite charges on the hydrogen and the oxygen, and that's a hydrogen bond. And then maybe define why is hydrogen bonding important? And like Jesse said, cohesion, boom, talk about cohesion as an example, right? So you can kind of hit what everyone said, hydrogen bond, polarity, and cohesion, and you just told that AP reader that you know a lot about water and a lot about biology. So I would stick with that. The second one, I know some of you are already moving on to the second one. Good. Javier says second one is polar covalent. Yeah, I would agree. I would say that the second model is really leaning into the idea that oxygen is electronegative. Yeah, it's showing polarity. Good, Peyton. It's showing that hydrogen is a partial positive, oxygen is a partial negative. It's just a really nice picture of polarity. The only reason it's not showing hydrogen bonding is because there's only 1 water molecule there, right? And hydrogen bonding, we're talking about 2 or more water molecules or 2 or more polar molecules interacting with one another. So hydrogen bonding would need to show another molecule interacting with that one. Okay, and the last one's a little obvious, but it would be your biological significance in explaining it. That would be key. So model 3. And look at the picture next to the molecules. Because yeah, you could definitely talk about cohesion. It is a picture of ice. But in this first picture, you could, like if there was no picture of ice and it was just molecules, you could totally talk about cohesion because it does look, it's just water molecules together. So a model of surface tension would look a little different. I mean, again, with each of these models, there could be 3 correct answers. With surface tension, you tend to see a model of a bunch of water molecules and then there being a clear surface on the top to show that that top layer, they're not being pulled at from a 360 point of view. They're only being pulled at from the left and the right and the bottom. So at the very top, it actually makes those bonds stronger to the ones at the top because they're not being pulled up. They're just being pulled up, toward and apart and then down. I would say model 3 is the biggest thing, is all about ice and how it floats. Okay, because normally when water is bonded together in liquids, it's not going to form these like hexagonal shapes. It's called like a lattice structure. And so what happens is when things are cold, remember the kinetic energy is lower and so it's slowing down. Okay, so when water slows down, those hydrogen bonds form and they're not going to be so easily like breaking and forming and breaking and forming. So you get this nice crystalline, or not crystalline, lattice structure. And that's what makes it less dense, okay? Because they're, in terms of like mass over volume, it's more spread out. The particles are going to be more spread out. Can you address density? Yeah, let's talk about, absolutely. So in this model, I would say it's talking about density. Yep, you could talk about density and how the tops of like freeze. Absolutely. Yep, talk about that. Well, very, very good. Yeah, so Peyton said, in her response, she would talk about density and how ice is less dense than water, which is really rare. So absolutely. And then remember, biological significance of that is that everything that is living in these aquatic environments, they're going to be able to survive when it gets cold. Otherwise, ice would just plummet to the bottom. So ice is a nice insulator at the top for those ecosystems. Will there be a live stream breaking down the structure of the AP Bio exam? I think someone did that on the very first one, talked about it a little bit, but we can absolutely do another one. And actually, because we're running out of time and I have so many more slides to go over with you, this is only the second question. Here's the third one. I have about 9 questions to do. So if I did another, like a follow-up to this, would you all be interested in like part 2? And we could just finish this potentially tomorrow night. You guys want to do that? Okay, awesome. Perfect. Yeah, because I do want to go over all this with you. You just had all really good time, really good responses. Let's do 8 o'clock again, because that's usually when they schedule my live streams. Because I do want to have you guys go through all this. I think it'd be really helpful. Ooh, Javier, is that too late or too early? You, the California person. 8 o'clock Eastern. Oh, you have practice tomorrow. Yeah, it'd have to be tomorrow because we start Unit 2 on Tuesday. How about we do, let's see if we can do 9 o'clock Eastern, because it would probably be a shorter one. It'd probably only be like 30 minutes. Tucker has work. Oh no. Well, Tucker, you can catch the replay if you can. Is that cool? 9 could work for most of you. Okay, let's do 9 Eastern. Awesome. It'll be short, so like 30 minutes maybe, but we'll just jump right in right here. I have about 10 more free response kind of questions, short free response, and then we'll open it up to any other questions that you guys have. Okay, awesome. Thanks so much for participating. It really, really is helpful that you come with questions. And I am going to come up with a way, maybe I'll have like a whiteboard or something so I can draw things out rather than writing it on a piece of paper and showing it to you. Okay, I will get that scheduled for tomorrow night. So hopefully I'll see you all then. Feel free to share this with your classmates too. I know you're probably all in big classrooms with other kids. Could we do a Kahoot? We could look into doing a Kahoot. I've never been a huge fan of Kahoots. I don't know why. I think I don't know. I like free response, open-ended questions. I think it makes you think a little bit more rather than just choosing A, B, C, or D. But maybe I'll add in a couple more multiple choice questions. All right, everybody. Thanks again for coming. We will see you tomorrow night. All righty. Oh, and let me show you if you want to find my schedule. There it is. There's my Twitter handle. So if you want to follow me on Twitter, I'll keep posting my schedule and when I'll be on here. All right, everybody, thanks for coming. Good luck at school tomorrow. Happy Monday tomorrow. I'll see you later.